Hello, happy Tuesday. Tuesday indeed. Jules, Salty Boy, Swam, Nana, Lucy Goose, Ethan Full, Marcus, Remy, Melissa. Hello, hello. Dale. Quack, quack, duck, attack. Damn, jump right in. Well, why not? The little intro. We'll do that intro when the time comes. Good to see you all. I was going to stream yesterday, but got carried away with stuff, and I didn't. So now we're here. Zio, hello. Kaba, good to see you as well. It's a beautiful day. I've had a lovely time. The weather in California is pleasant, and it feels like summer. What seems to happen is there's about 3 days of fog, and then there's about 3 days of sun, right? That's my observation. I was also reading a book about it. It's been gorgeous, yeah, Dale. It has been. It's been quite lovely. Where's the dragon tree? Dragon tree? It loves the sun, and it started to get some weird morphological changes because it hasn't been getting enough sun. So I have to keep it right by the window that's south-facing. So it gets a ton of south-facing light, and then the west windows. So it's south and west. It's right next to me. It's right here. Let me see if I can. You can see the leaves. Dracana Draco. Getting tall. But I want to make sure it grows nice. I was reading a book on the weather of the Bay Area. And I think I've told you this before. What kind of changes were happening? In the plant? The morphological changes. Let me see. I'll show you. So, if you look at the leaf morphology, the bottom leaves are the leaves that grew when the plant was in full sun. So what happens is the plant will develop its leaves under that condition that it is exposed to as the leaves are growing and developing, and then it moves on. When it moves on, it's always the new leaves that are continuously receiving stimuli and information from the environment and growing according to that environment. So the bottom leaves are real big, thick, and green because it was in full sun. And then winter came, and then periods of not as much light, and the leaves became a little bit smaller. So these leaves are not as thick, not as green. They're a little bit smaller than the other ones. So I want to make sure that they get enough light. It could also be a nutrient related thing, or it could be related to being pot bound, root bound, which could be the case. So we're gonna keep it in the light, give it a lot of sun. Hopefully it turns out right. If not, then we'll have to change the pot. It has gotten notably taller. I think it's gained like a whole 4 or 5 inches of stem. But I was watching the video of the dragon tree video I made, where I said, this is the Dracana Draco or whatever, and I put it in my car. I was like, I don't think it would fit in my car now. So we'll put it back. I got a new plant the other day. Actually, okay, it's gonna be a surprise. Do you know what this is? I have the label, but you can't see it. Let's see if it focuses. Do you know what this little thingy is? Little strange, right? A potato. Close. It's in a genus where the plants are sometimes called potatoes. Ant plant is a great guess. Lols. Is it a touch me not? Good thinking. But it's not. Okay. Ant plant, they both have swollen stems. Ant plant has a swollen stem. This has a swollen stem. Those are close. That's a surprise. The surprises sting the most. It is in the genus Dioscorea. And if you know, there are 3 things we generally associate as like potato-like things that we eat. There's the white potatoes, like russet potatoes. There's the sweet potatoes, and then there's yams. Yams are in the genus Dioscorea. Dioscorea. So this is related to a yam. And this one is from South Africa. It's from the arid lands of South Africa. And when it gets big, it looks kind of crazy. Tail was kind of bright in a roundabout way. Yucca. Yucca? No. Yucca is in the genus Manihot. But you can eat them. Good thinking, though. So let me show you what this plant looks like when it gets bigger. This is a baby one. And the cool thing about these plants is that I don't have a lot of growing space, but I do have a ton of sun in the windows. And this thing takes forever to grow. And it's cute. And it is an example. The elephant's foot, that's right. You got it. Dioscorea elephantipes. Elephantipes. I think it looks more like a tortoise. I would call it the tortoise. I would have called it Dioscorea trottoir or something like that. I wouldn't call it elephantipes. When you look at this, you're going to say, wow, they really dropped the ball with elephantipes. Hat. Your hat? Oh, wow, my face. Okay, here we go. My hate is off to you. Yeah, the hat. This is it. Dioscorea elephantipes. Look at that. This is what it looks like when it gets real big. The caudix, or the swollen stem, it's kind of tortoise shell looking. It's an example of a plant that drops its leaves in the summer. And then when it rains, it regrows them. It really is. It is something. Something cool. So I don't have a lot of space right now. And so I was like, well, what can I do? I can get relatively slow growing rare succulents and keep them in my extremely sunny hot window. And I can keep it in this pot for probably 5-10 years quite easily. And then it'll just look like one of these, like, you know, eventually. But it has leaves right now. We'll do a leaf watch. We'll watch and see how long it is until the leaves fall off. It has extremely shallow roots. I've jostled it a little bit and I can feel it doesn't have a lot. Okay, yeah. I'm trying to get the small kind of rare caduciforms. So I have this one, the Cyphostemma. It just gets a really thick stem. And then this one has a thick stem. So right now I'm on my my caduciform habit. Pam Smells, hello. What do I do for my hair routine? It's always spiffy. I've had the same hairstyle my whole life, and I tried the middle part. It didn't really feel right. I put product in it. I put oil. I comb it. Where did I purchase a new one? The Dry Garden. You know that one, Dale? I went there 10 years ago. And I went again with Marcus recently. This is just a eccentric, beautiful array of plants. Of course you do. We got to see Richard. Richard is the guy who owns a Dry Garden. He was fun. It's always interesting when people think you don't know anything about plants because they try and explain everything to you. And, you know, you have to humor them. When in reality, sometimes I feel like, is he gay? I don't think he's gay. He has a wife, right? I always see him with a woman. Maybe that's not his wife. Anyway, it's interesting when people share plant information. Oh, that's not what I was going to do. Plant information. And then you have to humor them. Sometimes it feels like the Ron Swanson meme. Where he's like, hey, I know more than you. I don't know if I know more than that guy, but sometimes when people tell me things, it's fascinating. Anyway, you thought he was there with this other man? The like Filipino guy that works there? I don't know. Who knows? I don't know. I think he's just a Berkeley straight guy. Berkeley is filled with eccentric characters. So, anyway, the dry garden. I was looking at the native habitat. I wanted to see that. Where was it? Oh, yeah. I want to see what it looked like. It's the arid lands. Classic Berkeley. I know, right? The arid lands of South Africa. We love a kind of straight. We love gender fluidity. Drex, hello. Brandon on YouTube. We are live on YouTube. Talking about plants, and we're going to watch a video here in a moment. Yeah. Byron. Hi, YouTube! Yeah, we started streaming on YouTube as well. More friends. There's some people who are really comfortable with live streaming, watching live streams on YouTube, so the more the merrier. Yeah, the old YT. In recent days, I've spent a lot of time thinking about native plants, which is fun. I love a native plant. Oh, hello, we have a new friend. Simpedo. Hello, welcome in. Thanks for subscribing. We'd love for you to join our Discord if you're interested. I spend a lot of time thinking about native plants. And whenever you mention native plants, people are always very opinionated. And it's always super fun to see everyone's takes on things. And, you know, in general, you always want to plant things that are native to your area because it'll benefit the native animals and plants pretty heavily. It's going to do really well in your climate. And there is some benefit to planting well-adapted ornamental species. We used to call them non-natives. The city of Austin, when I worked with them, they said, well, let's not call it non-native because that is like a social component, implying that just because it's not here, it's not from here, it shouldn't be here. So when I worked with the city of Austin doing plant education, they promoted the term well-adapted. A well-adapted species is a plant species that can do really well in the area without being invasive. And often those are horticultural varieties that have been cultivated for gardens. Simpedo, g'day mate, are you on Australia? What part of Australia? I've been looking at maps of Australia and the climates of Australia, and I'm so interested because it has all my favorite biomes or ecosystems. A lot of, I love the desert. I love tropical regions. It has Mediterranean climate. My three favorites, desert, Mediterranean, and tropical. Housewarming party this weekend. You got succulents. Darien, nice. You moved to a new place? Still in Sacramento? Nico Mouse. Did introduce species not work for the old Texas job? No, the city of Austin wanted, we could call them introduced species too. The city of Austin wanted to talk about them as well-adapted species. Sydney. Okay, isn't Sydney Mediterranean though? Duskborn. You want to see the seal? He's been sleeping for about 12 hours. Ta-da! He's back. This is my friend, the seal of approval. I saw a picture and someone said this is the seal of disapproval. That's crazy. Do you approve of Murashigi and Skoog mix? Okay, seal of approval. Do you know what that is? He does not know what that is, so we're going to have to spend today helping him understand what these things are. MS Media. MS is also known as Murashigi and Skoog. Oh, wow. Simpedo, you spent time in Namakwa-land, Africa. Deserts would bloom with carpets of flowers. It's amazing. They're in a cluster and better. Oh, Darren, you could keep them all clustered together. You could keep them clustered together, unless you feel like they're spilling out and competing too much then I would leave them in the same thing. It's probably a cute little arrangement. And the good thing is a lot of succulents don't have a lot of... they don't take up a ton of root space, so they can handle it pretty well. The seal of approval is here, and we will have to educate them. By the end of stream, remind me to make sure that the seal can approve or disapprove of it. They're in a cute arrangement. Okay then, leave them. That's perfect. Yeah, leave them be. But yeah, today we'll talk about tissue culture. Oh my gosh, I lost my thing. I googled away from it. Hydrate for plant. Ooh yeah, it's a good time to water some plants. Oh, I'm live on YouTube. I opened YouTube to find this video, and it popped up with a picture of me. Quite meta. Maybe I can watch my own live stream. Can we do that? Oh, that's crazy. Look at this. To find this video, and it popped up with a picture of me. That's a picture of me. Meta. Maybe I can watch my own live stream. We are watching our own live stream. Oh, that's crazy. It is crazy. It's okay, Matthew. To find this video, and it popped up with a picture of me. That's a picture of me. Meta. Maybe I can watch my own live stream. We are watching our own live stream. Oh, that's crazy. It is crazy. It's okay, Matthew. There's three. Look, I have the picture of me. That's a picture of me. Meta. Maybe I can watch my own. This is gonna be mirror dimension. Doctor Strange live stream. Oh, that's crazy. It is crazy. It's okay, Matthew. There's three. Look, I have the picture of me. That's a picture of me. Meta. Maybe I can watch my own. This is gonna be mirror dimension. Doctor Strange live stream. Oh, that's crazy. It is crazy. I think this is hell. It's okay, Matthew. There's three. Look, I have the. It is hell. Okay, we're not gonna watch that. Doctor Strange thighs seem thicker than usual. Well adapted Matthew invasive species. That's mitosis. Let's get to the thing. Anyway. Oh, it's already pulled up. That's what happened. Anyway, we're gonna water the plant. Oh, that little guy we just bought. Mitosis. Meiosis and mitosis are tearing us apart. Don't you realize I don't have a heart? How far would that have looped? Indefinitely. Actually. That's a Matthew for everyone. Macusis. Oh. Let's find this. I have not watered this guy yet, so we're going to water him with my own water. It's like in Dune. Have you seen the movie Dune? Water is so sacred. It's either me or the plant, and we share the same amount. Perfect. Drip, drip, drop. Okay. Things are just getting wet. Yeah, that's okay. It works out. Okay. Sufficiently watered. Green Monkey. Brandon. Hello. Welcome in. One of the things I was saying earlier. Oh, Diamond Buster. Is it possible to graft? Okay. I thought you were asking a plant grafting question. Good night, Salix. Is it possible to graft your mustache hair onto your mustache and then grow the ideal mustache like a apple tree? I don't know. I don't know how hair follicle grafting works. Presumably, it wouldn't. I would guess it wouldn't be successful. Ariana Grande is here. Hello. I was saying earlier, I've been thinking a lot about native plants and how wonderful they are. And their inclusion throughout our landscapes is critical for native creatures. And you can help native creatures by giving them more access to food. And there's one video that I thought was so exceptional, and so I would like to share it with you. This guy is a botanist in Utah, and I think he very properly and concisely describes this. I don't know how to make this bigger. Let me see. This plant is native to North America, but it's not native to Utah. Where is this?
America, but I still think it's really awesome and that it plays a super valuable role in our urban areas. There are very few species I would say that about, but Campsis radicans is one of them, and there are a few reasons why. The number one reason I like trumpet vine is that it provides an ecosystem function that no Utah native plant can. It is able to turn streetlights and telephone poles into massive hummingbird feeders thanks to its big red flowers and vining habit. There's just no Utah plant that can do that. It is equally important that this plant is not invasive here, and I've never seen it competing with native vegetation or getting out of control in natural areas. Also, Campsis radicans is native to the eastern United States, so it's not even that exotic here anyway. This is all unusual because, as a rule, there is always a native option you can plant in place of any given horticultural exotic that would be better suited to our specific climate and have more pollinator and wildlife interactions that contribute to local ecosystems. I think people often misunderstand championing native plants as being some sort of subjective preference or as if you're rooting for your favorite sports team or something, but that's really not the case. I prefer them because of the very specific set of ways that they exist on the landscape and interact with the world around us. It's not just about the label, it's about what the plant actually does. In this case, even though trumpet vine is non-native, it is filling a vacant niche, and I'm confident that they provide a massive boost to our urban hummingbirds foraging around the city. Also, I like it because I remember growing up in Salt Lake and always going with my friends trying to find flowers to suck all the nectar out of, so maybe I'm just nostalgic. Or maybe I'm a hummingbird.
So, I really appreciate that video because there is a clear and demonstrable example of a plant that provides, fulfills an ecological niche that is lacking, especially in built environments. So like in cityscapes and city spaces, sometimes the things that were once there that helped the hummingbirds are no longer able to provide those resources, and they can extend the like foraging seasonality, the access to nectar and food can be extended by these particular plants. And so I think that is a good example of how sometimes if there's a lot of people who are native purists who are like, if you plant anything that's not native to your area, you're the devil. And that's an outrageous statement. Then that is like this strange absolutism that lacks any nuance. And so this one plant is not native to Utah, but it fills that ecological role. So it gives extra food to hummingbirds. You know, fantastic. We like that. Your mom had the most magnificent trumpet vine you were a kid. Brixbury, hello. It's a pretty plant. Yeah, I think a lot of y'all know this one. I think it might even be native to Texas as well, but definitely eastern United States. You love trumpet vine on the East Coast. It's covered in bees. Yeah. Thunderbull, hello. The ending was awesome. Yeah, Brandon, I do agree. It's quite lovely. But yeah, so something to consider. Additionally, when you look at built environments, like I think a lot about street trees. In those specific street trees in San Francisco, there's many of them that are native, many of them that are not native, that are well-adapted species, and many of them are cultivated varieties that do really well in the city. And so like living in a city is very different from living in the native ecosystem and habitats. Like if you look at the tree, let me look this up, madrone. There's a tree called madrone. This is a beautiful native Californian tree. Let's see, Pacific madrone. It has this lovely peeling bark. Let me fix my face. It has this lovely peeling bark, beautiful foliage, beautiful roots. Provides a lot of ecological benefits. But if you plant it in cities, it doesn't do very well. You can see the native range here. So I'm thinking about San Francisco. It's Pacific Northwest, Northern California, and then little spotted parts of Southern California. Jules, okay, we'll do that in a second. If you plant this particular species in San Francisco, it has a somewhat challenging time because the city has its own series of problems with heat and soil and all that stuff. These native species often are susceptible to disease and pests, and so then people breed things that will succeed really well in urban environments. So there's a distinct, I saw some botanist also explaining this, and I heavily agreed with it, is that there's a distinct difference between the built environment garden and the ecological like systems that are native to that zone. Your 10-foot by 20-foot yard in San Francisco, you can do your best to make it as endemic as possible, which is lovely. And the reality is that you might have a hard time because of the microclimates created by the built environment. And so you might have to use non-native or species or cultivated varieties of native plants that are not truly wild, but they're garden varieties of the same thing. So this tree, you might have a hard time growing in San Francisco, but there is one that does super well. The strawberry tree. This is it's in the same genus, and it's a hybrid of the one we just saw. Tell me what you know about Japanese knotweed. It's not something you want. A horrible invasive plant. Yeah, no, it's a nightmare. I don't know much about it except that if you live in the temperate regions of North America on the eastern part, it's going to be obnoxious. I haven't had to deal with it. I've had to deal with other invasive plants, but this cultivated variety here is not invasive, and it's a hybrid between two different arbutus. It's a native arbutus to California and an arbutus from like Europe, maybe Ireland and Spain. And it benefits from something called hybrid vigor. Like when you grow plants that have really different genetics and you breed them together, they create a plant that is just super robust, super healthy, is notably quite large. And we often talk about it in the sense of corn. So here's an example within corn, but it's throughout our garden plants as well. There's the corn A, corn B, and you breed them and you get something that's a hybrid that has some crazy, huge fruit infructescence. The quality of the fruit is better, the plant is better, it's more resilient to disease, it's stronger. And so that's something that happens with the strawberry trees. Dale, you've been teaching many of your colleagues who just started a job at strawberry trees. Nice. Well, if it's Arbutus marina, then you can tell them this is a cultivated variety that's very much likely a hybrid of the native California arbutus and then the European arbutus, and it benefits from something called hybrid vigor. And so this applies to most living things. If you have two very genetically distinct parents, the offspring of those parents is going to have some fascinating traits that benefit it overall. Yeah. Thunderbolt, if you have inbred dogs and you mix the genetics, I think the same thing would occur. Wondering if it's similar with mixed dog breeds, slightly live longer. Yeah. On YouTube, our friend Omar God W7G. Your phone is watching you. You're planning what succulents you're going to plant, and you got this live feed recommended. Okay, well, if you got questions, I would love to answer them. Brandon on YouTube says, yeah, it's cost a fortune to deal with. Took over your backyard. Yeah. There are plants that are absolutely horrible invasive plants. Weed risk assessment. What is it? Australia. We have our Australian friend here. In Australia, weeds became so bad that there was this thing called the weed risk assessment that was developed, and it was adopted in Hawaii. The state of Hawaii, or kingdom of Hawaii, if you will, is the place on earth that has the most invasive species. And they have a whole department for invasive species. I took classes on, invasive species and weed mitigation and how to, prevent them and use integrative pest management to improve habitats and agricultural land. And so one of the things that is important to note is that if you have a plant that is not native to your area, there's a chance it could mess up your ecosystem, like Japanese knotweed and English ivy and all these different things. And so there's this assessment. Let me see if I have it somewhere. There's a PDF, but basically you fill out this big PDF and it gives you a ranking. This might be it. Give me the PDF. Or no, it's not a PDF. I'm sorry, Excel spreadsheet. And it has so many categories. And it says, hey, does it produce... Oh, here we go. No, these are all the plants. Anyway, it has like 50 rows. And each of those rows says, does it produce a lot of seeds? Does it disperse by the wind? Does it have dormancy in its seeds? Do the seeds stick to things? And it's completely ecologically robust. And it gives you ranking from 0, meaning probably not a problem, to positive numbers, meaning be careful, negative, meaning it's not going to be an issue. Positive, anything above like a 5 would be like, oh no, don't plant that. Don't use that. And different plants can be invasive in different areas. So there's a plant that's used all over the place in Texas and in North America. What is it called? Heavenly bamboo. Do you know this one? Heavenly bamboo. It's objectively a beautiful plant. There's nothing I can say about its morphology that would make me despise it. I think this is truly beautiful, and it should not be planted in Texas because these little red things are little fruits. The birds love to eat them up, and they love to go shit in the woods. And when they poop, they grow in the forest, and then they're everywhere. They take over. Isn't that crazy? Now, it's not invasive in California because it's not wet enough. So people can plant this in California and it doesn't spread and all that kind of stuff. It's too dry, and California doesn't get as much, precipitation throughout the year. But in Texas, you don't do that. And then there's also probably cultivated varieties that don't produce fruit. And so you can grow this plant without it being invasive. So that's one of the benefits to having cultivated varieties. People can breed these things. It's almost as if nature encourages diverse breeding. Yeah. I think that's genetic diversity is a point of sexual reproduction. Yep. And the thing that is accentuating it is in the example that I showed you. What they did was they took inbred corn to get a homogeneous or basically like identical genetics. So these two are completely inbred to the point where you're going to get the seed is going to have the same genetics throughout. And so that obviously is inbreeding isn't really an issue in some organisms the way we think of it in humans. Anyway. Okay. Jules requested we do some bicycle exercise, and then we'll watch this video on tissue culture. Jules, you're going to do it with me? I know it was a misclick, but unless someone can find a way to refund that. Are you going to join me in doing bicycle kick things? I've been using this cloak of levitation. Keep me happy.
Move like they heard.
Okay. You're gonna join me in this one too, lols. Oops. A misclick again. The cloak is so long. Put him to work. I already, I've been running around every day and laboring. My cloak makes it easier. I didn't want to reuse. I didn't want to use new shorts. Strange squats, I know, right? All right. Let's see. Have you seen this lady's videos?
Imagine being able to clone any plant hundreds or even thousands of times, all from just one single leaf. Well, I'm about to make your dreams come true because it is totally possible and not even that hard to do. Today I'm going to show you how to do plant tissue culture at home with an inexpensive DIY setup. No laboratory experience or prior knowledge required.
Okay.
All you need is a plant and a dream.
So
If
you
you're
can
not-
follow along at home. Maybe we'll do this. We'll, I don't know what plant I would want to tissue culture, but at present, maybe by the end of this we'll have some ideas. Plants in jars, one of your favorites. Okay, yeah. It's pretty good. We're gonna watch your video. Okay, so this is a, 45-minute how-to video for growing tissue culture. Basically, you take a small piece of a plant and you get it to differentiate. Well, you get it to become like a blob of cells that have no real identity. And then you give the cells an identity by applying growth regulators, and then it grows. Okay, let's watch.
Familiar with me, my name is Lore. I make plant tissue culture videos on YouTube. Here are all the places you can find me if you thirst for more microbiological content. I also have a new website, plantsandjars.shop, where I sell all of the chemicals that you need to start cloning your plants at home. On
You can buy all the stuff. She has kits, very convenient. Oh, Lord have mercy.
website, I also sell some really dope merch, which I'm modeling today. The back has this really cool skeleton doing tissue culture.
Cello, growth regulators are basically like plant hormones. They're chemicals that signal to the plant different stages of growth and development. It's like we have cortisol and serotonin and testosterone and estrogen. And plants have auxin and cytokinins and gibberellic acid. These are just compounds that tell the plant to do something when they receive a certain amount of them at a certain time in a certain place. In tissue culture, it's auxin and cytokinins. Well, let's see how she does it.
And I think after this run of shirts, I'm probably going to retire this design. So if you want it, make sure to pick it up. Buckle in because I'm pretty sure that this video is going to be the most thorough tissue culture tutorial to ever exist. What was that? You don't want to do tissue culture? You just randomly clicked on this video from your YouTube recommendations? Well, it's too late to turn back now. I'm an NPC who hands out side quests, and you already accepted this one. Unlike humans, plants are very drama-free about reproduction, so they can reproduce two ways, sexually and asexually. Tissue culture is a form of asexual reproduction, which means that the plants produced through tissue culture will be genetically identical to the plant that was used for the initial tissue sample. This is why tissue culture is sometimes referred to as cloning. We're literally making clones of a plant.
Magnificent, aren't they?
Every plant is a little bit different, but no matter what plant you're trying to clone, the first step is to find a good protocol. A protocol is a set.
So what plants would you want to use tissue culture for? Well, you would probably tissue culture something where you need to have identical copies of it. Perfectly identical copies because they're all genetically identical. This tissue culture is used a lot with specialty houseplants that have unique mutations to get a lot of them. In agriculture, it's used for plants that have special genetics like disease resistance. In Hawaii, there's a banana that they started to tissue culture that had disease resistance to certain types of fungi that were giving rot. There was a lot of rot in the bananas. And they said, wow, this is going to kill all of this type of crop. Let's create disease resistance. And they needed to do it pretty quickly. What they could do is take that banana and grow it in the ground, get it real big and divide up the corum and grow it that way. Or you take tiny pieces of cells and then you have hundreds of them. So then you can distribute that to the farmers. So, there's a lot of reasons why you might want to do tissue culture. Additionally, it's for conservation. You can keep these plants in these containers indefinitely. One of the great things about tissue culture is that you can have basically Noah's Ark. So if there were a horrible environmental disaster, you want to keep plants to repopulate the earth. You can keep them in a couple different forms. You can keep plants as seeds, but some plants don't stay as seeds for very long. And those plants would be called recalcitrant. The seeds will basically rot and go rancid after 6 to 24 months. And so how do you keep those plants? You can keep them in a botanical garden outside, or you can keep them in tissue culture. And all you have to do is constantly change out the media every year. Every year, all you have to do is just take the little tiny plant out and put it in a new container. Use radiation to induce mutations, and then tissue culture variegated plants. Infinite money glitch. Yeah, you could definitely do that. People have done that. That is a master's thesis for a horticultural master's. Yeah. So you could use tissue culture for conservation. You could use it for propagation, for industry, for houseplants, for agriculture, that kind of thing. I personally find the conservation component the most interesting because you basically have this Noah's Ark of tiny baby plants. Dale, take care. You're going to an open mic. Good comedy, hopefully, yeah. See you later.
Instructions for tissue culturing a certain type of plant. This is what a typical protocol looks like. They usually come out of research institutions. I use a free website called ResearchGate to find most of the protocols that I follow.
ResearchGate is pretty good. That's a good one. It's not like Elsevier, those greedy bastards. Okay, so lulz and Duskborn, we're talking about radiation for plants. This was a big thing until people started to get worried about being killed by radiation. It's called atomic gardening. So what people would do is hey, I'm going to create some really cool plants, and you can join the Atomic Gardening Club. What you do is you expose a bunch of different plants to gamma radiation. What they typically used was cobalt-60. It's a radioactive isotope of cobalt. And so they created disease-resistant peppermint plant using this type of method. There's a bunch of plants, and it's a whole cultural thing too. Post-war society in the 1950s, I think it was the 1950s, mutation breeding is the term people use for it as well. Exposing seeds to random mutations to try and get the desired outcomes. But yeah. Let's see, atomic gardening. Let's look that up real quick. Yeah, Duskborn. Have you followed Rusty Exotics? He grows orchids from seed, and it takes basically 2 or 3 years to get anything. So, yeah, you can definitely tissue culture them. When I was in Hawaii, they would tissue culture anthuriums for ornamental research. Or research on ornamental anthuriums, and also used it for conservation. Look at that. Atomic energized tulips. Dwarfed. Okay. Anyway, this is a big thing people have been doing for a long time.
Search for a protocol, you would type in the Latin name of a plant in addition to the keywords tissue culture or micropropagation. To save you time from having to read dozens and dozens of pages of very dense research text, I already did it for you. I've adapted a whole bunch of scientific protocols into very simple 2-pagers. Here's a before and here's an after so you can see the difference.
That's great. That's very fun. I think what she's doing a great job of democratizing science and democratizing this industry. That in 2020, everyone wanted those Thai constellation monstera and all this stuff. Well, you could buy it and then tissue culture it and then sell those, and then you create more supply, and then demand goes down. But I like that she's doing this. This is fantastic.
I also quote my sources in case you prefer to look at the source material. All of the protocols from the Plants in Jars protocol library are included whenever you order anything from my website. even a shirt. I think I have 10 protocols in the library so far, but I have 2 that I'm actively working on adding probably this weekend. Conveniently, every single protocol in the Plants in Jars protocol library also works with just the 5 products in my starter kit. So if you purchase those 5 products, you'll be able to do literally any of the protocols. Now you can see my divine plan. Monsteras are one of my favorite plants, so today we're going to be following this Monstera protocol. You can see that the first thing listed is the type of explant that we need. The tissue sample that gets used for tissue culture is called an explant.
Okay, there we go. There's our key word, explant. The piece of tissue you use for tissue culture. That's when you said Rio Red. The red Rio, I call it Rio Red. Grapefruit, yeah, from Atomic Gardening.
Remember that word because I'm about to say it 10,000 more times. The most common types of explants that I typically see used in protocols tend to be nodes and leaves. Oh my god. Nodes and leaves. A node is the part of the plant where the aerial roots and the new leaves originate. Like I said, you'll see nodes used in tissue culture a lot across many different protocols for different types of plants. Nodes happen to be really great explants for tissue culture because they contain a lot of meristematic cells. Meristematic cells are undifferentiated, sort of like human stem cells in a way. So they each have the ability to divide and eventually form a fully formed plant. When we use an explant that contains a lot of meristematic cells, like a node, we can potentially end up with a lot of plants, which is the goal of tissue culture. That's actually like the basis of why tissue culture works. It's not magic, it's just science.
The tissue culture is also called micropropagation. And so this is like a key technique within horticulture to make more things or to propagate. You're just doing it at a small scale. So,
how to remove these nodes and clean them later, because the first step of doing tissue culture is actually going to be to prepare the media. The media is this gel that the plants grow in. Looking at our protocol, we can see that there are 3 media formulations listed: establishment, multiplication, and rooting. These 3 media recipes correspond to the different stages of the tissue culture process. Since we're starting
okay. Yeah. So these different things are the IBA is, oh, you can't see it, my face is in the way. In the protocol, IBA is a plant hormone called auxin. And auxin is the thing that makes plants start rooting. So when you buy rooting hormone, you're buying a synthetic form of auxin. So IBA is the plant growth regulator or the plant hormone that makes roots. So in the rooting stage, the media contains 0.2 milligrams of auxin, which means make roots. I don't remember what NAA is. Okay, Pantherus Dark has a question on YouTube. Can you help me grow a calamansi orange tree from seed? It's your grandmother's tree and had an orange from a seed that sprouted in a wet paper towel. Any advice? I guess my initial thought is typically with cultivated citrus, the seeds are often not the fruit. So if you grew that seed up, it might not be the same as the fruit you ate. It might have different genetics. For folks who don't know, this is what calamansi looks like. I only know this as a citrus that Filipino cuisine uses. I do love the colors. It's quite fascinating. Orange on the outside. Orange on the inside, green on the outside. You learn seeds will be different, so transplant. Yeah. You're in... Panther is dark. You're in... Oh. You're in Wisconsin. Is that right? Well, give it a go. Grow it up nicely. Let's learn together. How to grow calamansi seed. How to grow the calamansi from seed. Germinating it. Remove the outer pulp as if you're harvesting. Okay. This is WikiHow. It's surprising. Well, maybe it's not surprising. WikiHow is really good, actually, for plant-related things. It's gonna get cold here and not sunny. Okay. Well, let's make it work for you. Here's the link to this. Oh, I guess it went twice. Or once. Okay. Remove the outer pulp as if you're harvesting seeds from calamansi fruit. Fold them into this paper towel. Leave them. Once they're germinated, you can pot them up. There is a specific type of citrus potting mix. It's basically like a well-draining soil. So you could buy cactus media and use that, but sometimes you'll find bags that say for citrus and palm trees. Typically, they group them together. So plant them into that. They should start to grow. They'll want full sun. Okay, this is the thing that you're going to be worried about, Pantherus dark, is that they like to be where it's subtropical or tropical. Calamansi, I think specifically, is tropical, but citrus in general are from like subtropical Asia. So 9B would be like central Texas and parts of, California and Florida. You're going to want to make sure it doesn't get below 40 degrees Fahrenheit. It doesn't like being cold. And it loves the sun. So you can buy a grow light if you want. Grow lights. You could even go to your local plant store and see if they sell them. But I typically just buy shop lights. You can buy something that is like a full spectrum white light. This kind of thing right here would be perfectly good. Grandma said tree since 70s and she kept it alive. You don't know how? She has it in a pot in Wisconsin. Does she move the pot indoors and outdoors? I'm curious. Anyway, yeah, you can give it a grow light. Give it a citrus potting mix. You can ask your grandmother how she kept hers alive. And they might be able to give you some tips and tricks. Okay, slightly acidic. That's the other key part. Citrus is fast draining and slightly acidic. The easiest thing would be to buy one of these. Oh, hello.
Yahoo.
Yahoo. Ethan Co, thanks for subscribing. Appreciate it. It's sweet and it's sour. Calamansi? Calamansi is so sour. Are you talking about your grandmother or the fruit? Okay.
Very beginning of the tissue culture process, today we need to prepare the establishment media following this recipe in the protocol. Astute viewers may notice that the recipes for the establishment media and the multiplication media are exactly the same. This is pretty normal. A lot of times you'll establish the explant directly into multiplication.
Okay, my face was covering that, but they're the same. But yeah. Pantherus Dark, let me know how it goes. If you have questions, come back and stream and ask, or message me on Instagram or something. I might see. I'll try and check them. But have I seen the horror movie The Girls with All the Gifts? Never heard of it. I don't really watch horror movies. The world itself is too horrific,
Regardless of the type of plant that you're working with, the media is typically going to be made up of 5 ingredients. The first ingredient is distilled water, which is going to make up the bulk of the media. Sucrose, which is just sugar. I just use white processed sugar from the grocery store. Murashigi and Scooge, or MS for short. MS is a mix of all the macro...
Seal of approval is going to learn what this is. MS Media, Murashigi and Scooge. When I was in college, they had us take a class called The Plant Cell. And I said, I don't know, I guess we're just learning about stuff about plant cells. And that's basically what we did. We only learned about plant cells. The first half was we spent a week on each organelle and the plant physiology behind every organelle. Vacuole, chloroplast, plastid, all that kind of stuff. Ribosomes, ER, Golgi apparatus, nucleus. Mitochondria. But yeah. The second part, we learned about various functions that plant cells undertake. And one of the things we learned is that there was this guy named Scooge. Scooge was really critical for like the discovery of certain, I guess, what was it? He discovered something pretty significant. Let's see if we can find it. Cytokinins. Okay. Plants have little hormones, basically, and there's two really big ones. Auxin, which is often related to rooting, and cytokinin, which is related to development of shoots and leaves. This guy, his name is Folk Karl Skog. This guy was an Olympian. He's from Sweden. Yeah, his story is crazy. I like his mustache, obviously. He is from Sweden, did track in the Olympics, and then ended up getting a PhD in plant science, and then discovered cytokinins, or critical components of it. And so he and I think it was his grad student or colleague, Murashige, oh my goodness, get me out, created this mix of nutrients. This is the most cited paper in plant research, is his paper with Murashige on what you put in this gel. These are all the things the plant needs to live. There's major salts. It's crazy, frazzled. I like mustache bias. I do. When people say that they have felt a specific way about something since they were a kid, I get it. I've said this on stream several times before. But when someone says, "Hey, I think I am a girl," or if they're, they look like a boy and they say, "I think I should be a girl," or something like that, I completely get it. Because when I was a little kid, I said, I need to have a mustache, no matter what. Anyway. Trans rights. We love trans rights. Auxins. So, Lols, you said auxins are responsible for phototropism. Yes, auxin is responsible for a ton of different things, and auxins are often at play with cytokinins. So it's the antagonistic component of auxin and cytokinin. So it's the relationship between those two. And there's also abscisic acid and gibberellic acid, which also do the same thing. These different plant hormones interact with each other. Not me coming out as a sign mustache at birth. That's true. Anyway, this media you need nitrogen, so it has ammonium nitrate. These are all salts. So it's going to be a positive ion and a negative ion that create a salt, a crystalline form. Calcium chloride, you need calcium, you do need chloride, you need magnesium, you need sulfite, you need sulfate. Hoagland solution. Why is that one used? It's for hydroponics. Yeah, you need different things in hydroponics. I don't know why you would use... Yeah, one is a gel and one is for water, right? I think that's very simple, but there's nuance in it. You need boron, cobalt, iron, manganese, potassium, iodine. These are minor salts. So the plants have micronutrient needs and micro and macronutrients. Micro are these. This one is fun. Okay, say this with me. Ethylene tetraacetic acid. Ethylene diamine tetraacetic acid. We'll get there. Glycine. What is glycine used for? It's an amino acid. They put some amino acid in this. I don't know exactly why these are included. Thymine, vitamin B1. Okay. Anyway, get me out of this. It's stuck. Okay, there we go.
Micronutrients that plants need to survive in tissue culture. The media that I sell on my website, which is the one I'm showing right now, also contains Gamborg's vitamins so that you don't need to add them separately. The fourth ingredient, or ingredients sometimes, are plant growth regulators, which is usually shortened to PGRs.
Okay, she gets it. For me, if someone who does this is a silly pedantic thing. Arboreal octopus, talking about tissue culture, micropropagation, how to grow plants in little tiny containers and to propagate in small spaces. For me, this is a small nuance thing. For someone who understands plant physiology and growth and development, we'll call them plant growth regulators. For science communication, it's easy to say hormone because people know what that is, but they're not like hormones at all. They're quite different. But for analogy, we understand our bodies decently well, and high school and stuff doesn't teach plant science very well. And so we understand hormones more than plant growth regulators, so it's easier to say plant hormones. But anyway, she gets it. One of the classes like TA, that was a final exam question. You know, in 2 or 3 paragraphs, explain why plant growth regulators are different from human hormones.
The last ingredient that we need to include in the media is agar. Agar is a gelling agent that turns our media from a liquid to a solid. We're also going to need some supplies for mixing our tissue culture media. Just as a reminder, everything that I use for this entire video is linked below. Between plantsandjars.shop and the Amazon link, you can find literally everything you need to get started. The first thing that we'll need is a cheap magnetic stirrer and a stir bar, a 1-liter beaker, a gram scale that goes to 2 decimal places, pour boats for the scale, a micro pipette and pipette tips, a pH meter, pH up and down hydroponic solutions to adjust the pH, polypropylene deli containers, and either a pressure cooker or an Instapot. No matter what protocol you're following, whether it's one you found on ResearchGate or one from the Plants and Jars protocol library, the ingredient quantities
Unfortunately, she did identify. I'm going to give you a video that will allow you to do everything. You can do all of this at home. You don't need to get that much. But then the micro pipette is a little bit specific.
Will always be to make 1 liter of media. So if you want to make half a liter, divide everything by 2. If you wanted to make 2 liters, multiply everything by 2. You guys are smart, you get that. I start by adding 800 milliliters of distilled water to my beaker. Then I add the stir bar and turn on the magnetic stirrer. Although we're going to be making an entire liter of media, we don't want to start with an entire liter of distilled water. If we did start with a full liter of water, the ingredients that we add afterwards would push the water level up over a liter and we'd end up with messed up ingredient ratios. So we'll top off the water after we add all of the other ingredients.
Yeah, on YouTube, our friend Omar God said she has a list for everything. Amazon list. Yeah. Lols. Yeah, you can get cheap ones. It's just funny that it is necessary and very specific. This is for tissue culture, micropropagation, not hydroponics.
According to our protocol, the first thing that we need to add is 4.44 grams of MS. I measure out 4.44 grams on the scale and then I add it to the beaker. Next, we need to add 30 grams of sucrose. Just as a reminder, sucrose is just sugar, and sugar is going to be the energy source for our baby plants. Next, it's time to add our plant growth regulators, or our PGRs for short. There are 2 categories of plant growth regulators that I use very, very commonly: cytokinins and auxins.
Yes.
Cytokinins are plant growth regulators that promote cell division. Typically, we see a higher concentration of cytokinins like BAP used during the multiplication stage of tissue culture, when we want the plants to be multiplying very rapidly. Cytokinins help the plants to do that. The other category of plant growth regulators is auxins. NAA and IBA are 2 types of auxins. Auxins are plant growth regulators that induce root formation, so we typically see them.
I think I'm curious why she's using NAA and IBA, so I would like to learn that in this video. The amounts are different depending on the plant. Yeah, so I think what happens is people want to create, what happens is researchers create protocols and they do these large, like, there's a type of experiment where you just give a bunch of like concentrations in different amounts of things to see what works best. And I forgot the name of that, like, experiment. But people will basically figure out the best amounts for each plant and dose titration. It's kind of like that. And a NOAG, hello. It's kind of like that. Green bean stream. Easy to contaminate. Yeah, I think she's going to use a pressure cooker to make sure everything's sterilized. But yeah, each plant is going to have different ratios that it does best with based on the way that its internal physiology, operates. And all of that is pulled from research. So someone has done the experiments. Relo. Okay, sounds good.
Used in higher concentrations during the rooting stage of tissue culture. The solutions that I sell are all concentrated in 1 milligram per milliliter. This particular Monstera protocol calls for 5 milligrams per liter of BAP. So that means that we need to add 5 milliliters of the BAP solution to our media. I personally think that the easiest way to measure out very small amounts of liquid is with a micropipette. I purchased this one from Amazon for around $30. There are different sizes of micropipettes available. This particular one is for 100 to 1000 microliters. You might be thinking, what the is a microliter? 1 milliliter equals 1000 microliters. So that means that this particular micropipette can measure anywhere from 0.1 to 1 milliliter. I understand that.
That makes sense. I want to see this real quick. Okay.
During the rooting stage, milliliters, so it probably would have been a better option. However, if you're only going to get one, I would recommend getting the smaller one. But I'll link both below. Now I have 5 milliliters of BAP solution in my tissue culture media. Referring back to the protocol, we can see that we also need to add 0.1 milligrams per liter of NAA. To do this, I get a fresh pipette tip, set the micropipette to 100 microliters, which is equivalent to 0.
It's very precise. And one of the things is that plants are super sensitive, especially when they're this small. But plants are highly sensitive to the changes in growth regulators and everything else. Dascorn says a medium optimization for media screening trial. That's the very specific name for it. Yeah, that's right. Yeah, extremely precise. With a small volume, pipette can calibrate right will determine if it's accurate. Yeah, that's fair. And Noah, you know a lot about these micropipetting devices, huh? Lucy Goose, take care. I was curious why they're using two different synthetic forms of auxin, IBA and NAA. They're both synthetic forms that don't break down easily, and it looks like the big benefit is that they kind of help each other fill in the gaps. Basically, having these two working in conjunction ensures successful root development. You have a few personal ones, okay. Pipettes. The seal fell asleep. He's listening now. The seal is still here. Yeah, you're a microbiologist. You see a lot of pipettes. Serial dilution is one way to get to small concentrations. Oh, wow, yeah.
1 milliliters, and then I add that NAA solution directly to our media. The final ingredient that gets added is our agar, or our gelling agent. This protocol called
NAA is cytokinin, which is the thing that she identified that makes the plants divide. And then, no, BAP is cytokinin. NAA and IBA are the auxin. Brian, hello.
7 grams of agar, so I measure it out on the scale and add it to the media. Most protocols are going to call for between 6 to 8 grams of agar per liter. Anything outside of that range, be skeptical, okay? I personally think 7 grams of agar is the sweet spot for the gelling agent. Next, we're going to top off that water to the 1 liter mark and then take an initial pH reading. The pH is currently too low. According to our protocol, it needs to be at 5.7. To increase the pH, I use my micropipette to dispense a few drops of hydroponic up solution at a time until I'm close to 5.7. I've used a lot of pH probes in my lifetime. My personal favorite is this one from Opera. But this one is totally overpriced. Opera, if you're watching this is too expensive. So I will also link a more economical version below as well. Now we are ready to pour our media into our individual media containers. This is important. The media containers need to be made of polypropylene in order to withstand the pressure cooker, which is going to max out around 100.
The pressure cooker. Nice. Duskborn, you said, could these chemicals be used to eliminate unwanted plants? I think auxin can be an herbicide because everything is about the dose, right? Anything can be a poison if it's too much, right? Let's see, auxin herbicide action, lifting the veil step by step. Auxin herbicides stimulate a variety of growth and development processes when at low concentrations, but higher concentrations, they're uncontrolled fatal cell elongation. Let's see. Is there a picture? Yeah, basically it disrupts growth and development.
21 degrees
I use it for canning. Nice.
PET and other types of plastic will melt, so really make sure you're aboard the PP train. Also, watch your back because sometimes containers will be made of PP, but the lids will be made of PET or something else. While you're pouring the media, if you find that the agar and the sugar is settling at the bottom, just put it back on the magnetic stirrer and give it a good mix to redistribute the ingredients. And then you can resume pouring. If the ingredients are not properly distributed, you'll end up with some containers of media that are still liquid and some that are way too hard. Now that we have our media poured, the next step is to sterilize the media in either a pressure cooker or an Instapot. In scientific terms, this pressure cooking process is called autoclaving.
Autoclaving.
The purpose of autoclaving is to kill any sort of bacteria or fungus that might be in the media. The media is mostly sugar and water, so if there's any sort of contaminant in there, it will grow much faster than the plants and you'll have to start over. For some reason, I feel like people are the most hung up about this pressure cooker step out of all of the steps in the process of doing tissue culture. Yes, back in old times, sometimes pressure cookers would explode. Modern pressure cookers are built different, they're very reliable, and they tend to be really safe. So don't fear the pressure cooker, it's more afraid of you than you are of it. The pressure cooker that I'm using today is the 23-quart Presto pressure cooker from Amazon. I've sold so many of these, I'm like a pressure cooker influencer. Following the manual for this specific pressure cooker model, I start by adding 12 cups of water, and then I crumple up 3 balls of aluminum foil and throw them into the pressure cooker. The foil balls support the first pressure cooker rack. I think spacers exist, but to be honest, the aluminum foil balls have always just...
The foil is crazy. She could buy spacers or she could crumple up aluminum foil. Okay, hey, whatever works, right? She's got jokes.
So well that I've never even looked into buying spacers. This is street tissue culture. I can fit about 8 or 9 containers of media per level, so 24 to 27 total containers inside the pressure cooker. Of course, it depends on the size of your polypropylene or glass media containers, as well as the size of the pressure cooker or Instapot.
I feel like
Sorry, I keep forgetting this is an...
She does so many micro propagations that it seems like she would be saving money by having like a actual spacer. Would Jello work? Agar is a little different from gelatin. Jello is derived from collagen, right? Or like hooves or things. And agar is from seaweed. I guess I feel like it could... I don't know. Streaming beans at one time sounds very laborious. Steaming these one at a time. Yeah, I didn't think she was doing them one at a time like this. But she fits like 24 into a pressure cooker. Just put some ears of corn. Yeah, well. Cook something and eat it at the same time. Heartwarming. This chemist shows it only takes one passionate woman to collapse an industry. Yeah. Okay, if you bought a Jello packet, it contains acid that would harm the plant tissue, and it melts too quickly. and it would break down easily, and plant enzymes would digest the Jell-O protein. The melting point, okay, this is what it is. The melting point of gelatin is too low for most rooms. Agar stays solid at higher room temperatures. Okay, that's the big thing, is its melting point, I think. Pacifist run. This is how you make tamales, yeah.
Pot inclusive space. Don't snap the lids onto the containers, just leave them resting on top. If they are snapped shut, they will get really deformed under pressure and become unusable. PV equals nRT or whatever. After the pressure cooker is
Yeah, and a NOAG. Yeah, agar is more stable at room temperature as a solid.
loaded, secure the lid and turn the burner up to high. Once steam starts coming out of this hole, turn on a timer for 10 minutes. After 10 minutes of releasing steam, put the rocker onto the pressure cooker to start building up that pressure. Once the pressure is at 15 psi, we want to keep it there for 15 minutes. If the pressure is getting too high at any time, just turn the burner down slightly and the pressure will start to reduce. And once it's cool enough, I spray my gloved hands with 70% isopropyl alcohol, and then I snap each lid onto its respective container.
Nice.
Try to be very careful not to open the lids when you do this. We do not want to expose that media to any of the air in your house. No offense.
That media is going to be delicious for microbacteria, or microbiota. And then Anna Noack, you said, if I've ever had coffee jelly, yes, I love coffee jelly. Go get to the boba. Get some boba with coffee jelly. It is agar. Yeah, it's thick.
Using an Instant Pot instead of a pressure cooker. I know the Instant Pot Max does get up to 15 psi. If you remember my genetic modification.
Green bean. You were identifying the issues with the stuff about contamination. This is the whole contamination step. Ooh, I follow this guy. This is Sebastian. Yeah, I saw this tweet as well. Yeah, this tweet went crazy. I remember this one. He said his autoclave died, so he started using a pressure cooker. As much as you love tamales, you're traumatized because your mom and brother had to tried to make them once and gave 12 people food poisoning. That's crazy. For me, tamales are purchased from someone who makes only tamales.
Video that I did with Sebastian Kochoba, he uses an Instapot to sterilize everything. I've never used an Instapot. I just know it's possible, so I want to put that option out there into the world. In some of my videos, I have used a microwave to sterilize tissue culture media, and to be completely honest, I no longer recommend that method. I've learned a lot since then, and it's just really not that effective of a sterilization method, so sorry about that. My bad. Now that our media is sterilized, we also need to sterilize our tools. Anything that is touching the plants needs to be completely clean.
Yeah, that's the thing. So, Green Bean, you said all the people you know that have good tissue culture rates have labs. Yeah. The tools need to be clean. Anytime I've done anything related to this, has always been like special hoods, flow hoods. Everyone is sprayed with, she said, 70% isopropyl. I remember that smell. It's just like everything smells like really intense Everclear and vodka, and you can't. Breaking Bad, yeah, it's super you gotta be super sanitary.
What we're going to need today are stainless steel forceps and a stainless steel pair of scissors.
You like
I
the
wrapped
smell?
the forceps and the scissors in aluminum foil before placing them into the pressure cooker. We're also going to need some sterile water for rinsing our explants later on after we clean them. So I pour some distilled water into the same type of polypropylene containers that I used for the media. The process for sterilizing tools is exactly the same as sterilizing media. We want to sterilize them for 15 minutes at 15 psi. If there's room inside of the pressure cooker, you can include the tools with the media and run it all in one load.
So, for me, this protocol is only worthwhile if I need to clone something very special. If it is Monstera and it's not an extremely rare cultivar, I'm not doing micropropagation for a Monstera because I can do leaf cuttings and it will grow perfectly well. In this instance, I need like $1,000 worth of setup, and I have to sterilize everything. And I have to have the smell of a spontaneous nap everywhere, and yeah, I mean,
not room, then do 2 separate loads like I'm showing you here. Once we have the media and the tools sterilized, it's time to actually do some tissue culture. This is a still air box. We're going to do tissue culture inside of it. A still air box is just an enclosed container. There's really not much to it. The purpose of it is to prevent the air from moving while you're working with your sterile cultures, which reduces the risk of airborne contamination. I was sent the still air box a long time ago to use for videos. Thank you, Bellabora. You can also make a broke boy still air box by just
okay, still air box. This is a good way to prevent air contamination. Rare plant seem like a pyramid scheme. I mean, I don't really care for rare plants because often, why do people want rare plants? That's my first question. Why do people want rare plants? Do they just love the plant and it happens to be rare? Or do they want the plant as a status symbol? Or do they want the plant because it's like the thing to do? I get very frustrated with people who are plant collectors who are like, yeah, I need to get the euphorbia, whatever, because I need it in my collection. Well, why do you need it in your collection? Are you preserving it for conservation, or is this an ego? Is this like Jake Paul's Pokemon? I haven't seen it, but why the hell does he need all those Pokemon cards? I don't know. I get it. It's fun to have things, but like, I don't know. Anyway. Not sure about a pyramid scheme, but it feels like a scam. I think it's like a market thing. There's a company called Palm Street. Do you know Palm Street? I always feel quite suspicious of them. It always feels like weird. Palm Street. Does anyone have any thoughts on Palm Street? Because he's a pointless, uninteresting human being who's so purpose to make things work. That's fair. Monstera is a basic houseplant to you. It is, I think, the textbook example of a houseplant. Some genuses are worse than others. More specifically, some species more than... Yeah. This lady has so many plant videos about how it's a scam, and she'll plummet the price of rare plant. Yes! Marcus, great point. Like, I think this is one of those companies that feels like a trap. The plural of genus is genera. There we go. Yeah. They do these, like, auctions of plants. And they do these rare plant auctions. Let's see if we can click on it. Not clickable. Like, I met someone like a year ago who said, yeah, I got this like pink darling philodendron extraordinaire rare variegata from Palm Street. It was only $400. And I'm thinking, that is crazy. $400 for a specific genotype that might be morphologically identical, if not barely different from anything else. Talk about a company's individuals will ban her from purchasing because she knows how to reproduce them. That's fair. I think, Marcus, doesn't she even talk about Palm Street? Anyway. I follow these people on Instagram because they have pictures of plants, but it feels nefarious, and it feels like they feel like culture vultures, you know? What's that guy? Baby No Money? Speaking of culture vultures.
cutting 2 holes into any clear plastic container. First, we're going to spray down our entire work surface with 70% alcohol. Also spray the inside and the outside of the still air box very thoroughly with that 70%.
Yeah. Anna, thank you. I appreciate it. I typically say genera. I don't know why I said genuses. I think it's fun sometimes to say things in silly ways.
Don't be afraid to really stink up your house. When you go to the store to pick up your supplies, you will notice that they sell both 70% and 91% alcohol. 91% might seem like the better option because it's more highly concentrated, but actually 70% is better for surface sterilization. It takes longer to evaporate than the 91%. So it has more time to kill all of those microbes that we don't want.
That is a great little fact. I'd never realized. Yeah, 70% is better because it doesn't evaporate as quickly, so it can kill things because it persists. Better than 99%. Fun. I will be putting that in my pocket for when I go to the bar and need fun facts.
Goes into the still air box also gets sprayed with, you guessed it, 70% alcohol. Inside of the still air box, we're going to have some containers of the tissue culture media that we prepared earlier, our containers of sterile water for rinsing our explants, the sterile forceps and scissors. Leave all the tools inside of the aluminum foil for now, plastic wrap that's been cut into 1-inch sections. You can also buy grafting tape for this purpose, but it's kind of expensive. So I always just cut a roll of plastic wrap into 1-inch sections with a PVC pipe cutter. This is going to get used for sealing our tissue culture containers later on. And lastly, you need a...
I like the innovation with the PVC pipe cutter. I was like, I know that tool. That's for irrigation. Very clever. Dude, Dalalok, good morning. You haven't been streaming a long time. I've been gone for a long time. But yeah, Dalalok, good to see you.
Glass bead sterilizer. The glass bead sterilizer plugs into a wall outlet, so I run the cord through the side of the still air box. It heats up really hot to kill bacteria, and this is where we're going to place our forceps and our scissors when we need to put them down. At this point, we're ready to take our explants from our Monstera plant. Since our tissue culture plants are going to be genetic copies of this plant that we're using for the tissue sample, it's good to start with a healthy and physically attractive plant, or like physically attractive for a plant. I'm not attracted to plants. Just as a reminder from earlier, the part of the plant that the protocol specifies we should be using as our explant are nodes. This Monstera happens to have a lot of nodes. However, some of the nodes are very thick. We don't want to use these really large nodes for tissue culture because they're going to be very difficult to properly clean. Instead, we want to use smaller nodes like these ones we can see at the base of the plant. I grab a clean pair of scissors, spray them with...
Earthy nodes are not ideal. Stream of May, write that down. Visually attractive plant. Yeah, choose a plant that looks healthy, doesn't have like deformities and that kind of thing. There's a personal growth metaphor in the 70% alcohol thing. Too much force will coagulate your defensive walls or something. You cook? Okay. 70% alcohol, small nodes. Not too much alcohol, not too big. It's got to be just right. Yeah, I knew you use alcohol to sterilize things, but I didn't realize it was because of the evaporation rate. Yeah. Write that down. Put that on a t-shirt. 70% is good enough. Small nodes are better.
70% alcohol, and then I begin to cut my plant to harvest it for its nodes. Between each cut that I make, I'm respraying the scissors with 70% alcohol to keep them clean. While you're hacking away at your plant, try not to let it take advantage of your higher life form weaknesses like empathy. Your ancestors spent millions of years developing a brain capable of high-level problem solving and abstract thought, and they are just plants. Here are...
That was a crazy thing. Don't be seduced by empathy. That's a funny joke. Left Boy Games, hello. We're learning about micropropagation, how to make clones of plants at a microscopic scale using agar, human superiority. That's very medieval Catholic of her.
3 nodes we ended up with. Sometimes you can only get a few explants from one plant. That's why I don't always recommend making a full liter of tissue culture media. Sometimes it can just be way too much. For instructions on how to sterilize our nodes or our explants before they go into tissue culture, we need to refer back to the protocol. The protocol says to wash our explants thoroughly with dish soap under running water. You don't want to damage the tissue, but I really do give them a good scrub. Plants that are grown outside are always going to be more difficult to clean than plants that are grown indoors or in a greenhouse. After the explants are washed in the sink, the protocol gives specific information about preparing a sterilization solution, which is made of water, bleach, and dish soap. I combine 100 milliliters of household bleach with 900 milliliters of water.
That fun thing is that Dawn Ultra is like the standard scientific like dish soap.
And dish soap. I combine
Every dish soap I know in a laboratory is Dawn with a duck. If it's not Dawn with a duck, then what the fuck? That should be a t-shirt. Put that on a t-shirt and we'll sell that to laboratories. There's the duck. Where's Quack Attack? Is Quack Attack still here? Quack Quack Duck Attack. For cleaning animals. Yeah. Okay. You know, like, I don't know. Some people are new and they might not know this, but I really like the old Quaker Oats Wilford Brimley commercials. You know, Wilford Brimley was this old mustachioed guy and he did these advertisements for Quaker oatmeal. And he said, you know what? Every day I wake up and I eat a steaming hot bowl of Quaker Oats because it's the right thing to do. And he just kind of walked around. He's like an old cowboy. And when they hired him to do the advertisements, Wilford Brimley. Yeah, Wilford Brimley. He died like 2022. Quaker Oats. So I love his style of like, when he showed up, they said, hey, we have a script for you, Wilford. We got a cute story. We have a cute little story you can use, Wilford. And we're going to make the advertisement. He said, no, we're not doing that. I'm going to tell you how oats are good for you and they're good for your heart. And that's what they did. He just like ran over them. Anyway, we need that kind of advertisement for this duck soap. You know, if it's not Dawn with a duck, then what the fuck? We'll put that on the t-shirt. I'm gonna email Dawn and say, hey, that little duck is your mascot now. And ever since British Petroleum tried to kill everyone, they've been cleaning animals with Dawn dish soap. Yeah, Wilford Brimley. Used to know a DK Smash main with that tag, really? Wilford? Marcus, what did you say? Add soap. If it's not Dawn with a duck, what a fuck. Dawn dish soap is a great effective in labs and in the field. That's right. Okay. Okay, we have we have a command for soap. Show us my Dawn. What the fuck? Yeah, Green Bean Stream. It was British Petroleum that when I was in high school tried to kill us all. But the thing is, actually, Exxon's also trying to kill us all. Same.
100 milliliters of household bleach with 900 milliliters of water to make my 1-to-9 bleach solution. And then I add a small drop of dish soap as well. The drop of dish soap is going to act as a surfactant. A surfactant is something that reduces surface tension. So the dish soap is reducing surface tension between the node and the bleach, allowing the bleach to penetrate the node more deeply. These explants will be going for a wild ride on the magnetic stirrer, so I also add a stir bar. Now the protocol says that the sterilization time depends on the thickness of the nodes. Our nodes are fairly small, so we're going to be agitating them in the bleach solution for 20 minutes.
Yeah, the bleach thing, I kind of forgot about the use of bleach. We had a friend who had a question about... We had a friend who had a question about the Ducks and Dawn. Ducks and Dawn. Okay, who was that? Royal Havo. Wasn't it a marketing scam? I checked. According to the National Association... Or NOAA. What is it? NOAA? What does that stand for? National Oceanic something Association? NOAA, the government organization, said no. Dawn dish soap did not kill animals, and it was not a scam. The International Bird Rescue and the Tri-State Bird Rescue and Research Facilities genuinely did use regular blue Dawn dish soap to break down crude oil. Atmospheric. Okay, there we go. They actually used that exact blue Dawn dish soap to break down crude oil on birds and mammals. And so it did work. But, Royal, the animals did die because the oil is toxic. And so some of the animals did end up dying because of the toxicity of all the oil. Oil is just ridiculous. Okay. Okay, there's some notes about risk of home use before we get back to the video. People should never attempt to wash wild animals or domestic animals with Dawn dish soap. The National Atmospheric and Oceanographic Association. That's right. Stream of May, thank you. Okay, we have a quick public service announcement before we continue. Be sure not to wash wild animals with Dawn dish soap without professional guidance. It will turn them into dishes. Okay. Dumb advice? Last time a wild animal needed my help, I did use Dawn dish soap. And olive oil. You know there's those sticky traps?
One thing I think I forgot to film is it's good to tip the jar 180 degrees so that the bleach solution coats the entire inside of the jar. That can help maintain sterility. After 20 minutes is up, I spray the outside of the jar with 70%.
Okay. Unfortunately, she's not seducing me on this technique. This protocol requires way too many steps of sanitization, which I totally get for the science of it. I would only do this if it was worth it for me. If I'm propagating Monstera, I'm not doing this. Your convert to Dawn Platinum. Oh, it has alcohol in the formula. Oh, wow. Okay. Dawn Platinum? Really? You want to wash Jimothy? He seems like a gentleman. Olive oil is a beautiful oil. I love that little burning flavor of olive oil. Buff Boy Games.
And alcohol, and I place it into the still air box. Huzzah! We are now ready to establish our clean explants in tissue culture. My protocols give specific instructions for whether you're working in a still air box or a laminar flow hood. Since we're working in a still air box, we're going to follow the still air box instructions. Just as a reminder, here is where we are at in terms of the setup inside of the still air box right now. Before I stick my arms through the armholes, I'm going to wash my hands and my forearms under the sink with soap, and I'm going to put on a fresh pair of gloves and then spray the gloves with alcohol. If you have to take your hands out of the box at any time, you need to redo those steps before you put them back in. The first thing we need to do inside of the still air box is rinse the bleach solution off of the explants. I unwrap the foil that contains our sterile forceps and our sterile scissors, and I place the tools into the glass bead sterilizer. Then I open the container of bleach.
Yeah, I've worked in sterile conditions before, and there's a reason why I shifted from laboratory work to horticultural work. I like being covered in dirt. Thankful all the different way people exist. She has the ability to take what eons cultivate. Yeah, let me play in the dirt, exactly. I'm more of a, let me play in the dirt. Even though my background is in genetics and all that, that's why I left. It's too much. Imagine being the top 0.0001% most resilient germ. We have a friend from Chile watching. Hello, Mac. Your job is 100% lab work. Hey, it takes all kinds of people to do the wonderful things in our world. Yeah, Ananoag. All these protocol with the spraying of everything would make sense if you're doing it at home. Stream of May, when you were working with GMOs, okay, you had 1 minute. Did you submerge your hands in 70% ethyl alcohol for 1 minute and then 30 seconds in 1% bleach, and you washed in sterile water? It makes sense considering the nature of that work.
That contains our explants, and using my forceps, I transfer
you submerge the plants
explant into its own container.
Okay, that's good. I mean, that makes sense. It also kind of makes sense if you had to submerge your arms.
Sterile water. If we were working inside of a flow hood instead of a still air box, we would usually perform 3 rinses with sterile water. However, attempting this inside of a still air box increases the risk of contamination, so that's why I'm doing it this way instead, and I found it does work. When I'm finished with my forceps,
You mentioned a flow hood. If you don't know what I think many of you know what a flow hood is, but for folks who don't know what a flow hood is, it's this device that is a large hood, and I think what it does, it's a laminar flow hood that pushes air from the top and pushes it outward, so that there's always like air coming out of it, so that if you're breathing, your breath isn't going like into it, and the air in the room isn't flowing in and contaminating. It's constantly cleaning, filtering, and pushing clean, air downwards so that air can't come in. Alternatively, you could have the box with the gloves. This is only $2,358.75. That's not too bad. What about this one? Flow. I did like the flow hood because you always had a constant gentle breeze blowing on you. A curtain of air, that's a great way to put it. And Noah, you said there's a curtain of air to prevent dust and bugs and other things from falling in. This is why you use a biosafety cabinet. Protects the user. Okay, that's fair. Biosafety cabinet to protect the user, but if you're not too worried, you can use a flow hood. It's crazy how effective it is. It's super effective. Critical hit, even.
place them back into the glass bead sterilizer. At this point, each of our explants is floating in sterile water, matrix style. Leave the lids on the containers of sterile water when you're not actively handling the plants. Now, some of the tissue on each of the nodes has died from being exposed to bleach. So before these plants can get established in the tissue culture media, we need to remove the dead tissue. Dead plant tissue is a breeding ground for microbial growth, which could contaminate the cultures, so that's why we need to remove it. Any tissue that has turned white or brown or black needs to respectfully GTFO. Using the sterile forceps and scissors, I remove about 1/2 centimeter of tissue from both ends of each node.
For the record, Deskborn, we're only like halfway through the video. The good thing about this is I think 1/3 of what she's described at this point is basic standard sterile technique. Spray everything with the alcohol, keep those tools in the little beads glass sterilizer thing. Most of it becomes like part of the mise en scène. It becomes part of the regular routine of the space. And so everything she's describing, it's not as intense once you start doing it. If you want to be real dodgy, you've used a spray bottle, spray the air with 70% alcohol, and hold your breath. Interesting. Okay, and Anoag, you also had a note about grocery stores using that blast of air at the entrance to create this air curtain. Oh, I like it. That's a great point.
If you need to put down the explant at any time while you're working with it, put it back into the sterile water. After I'm finished removing the dead tissue from each node, I place my scissors back into the glass bead sterilizer, and I slowly grab a container of tissue culture media. I say slowly because you really don't want to disturb the air inside of the still air box, lest you defeat the purpose of it. We want each explant to be making good contact with the tissue culture media, but we also don't want it completely submerged in the media. After the explant is placed into the media container, immediately snap on the lid. You can theoretically put multiple explants into each media container, but I prefer not to, because if one is contaminated, then it can cause the other explant to get contaminated as well. Once I have all of my explants established in their culture containers, I wrap each container with plastic wrap to try to mitigate contamination even further before taking them out of the still air box. And there is our final product.
Okay, there
At
you
this
go.
point,
Look,
you can place
that's
your cultures under.
That's what it looks like. I've seen so many rooms filled with tissue culture. It's like, those little metal racks? They're just like 100 metal racks with like 400 or 800 grow lights. And the room has this crazy bright LED glow, and there's like little tiny baby plants. It's kind of cool to see, I kind of like it.
LED grow lights. I keep my lights on a timer, so they are on for 16 hours a day,
Did I
and
do
then
that?
they go off
Oh, Jules,
for
you
8
did.
hours at night. The lights that I use in my laboratory are from Horty Power. They're the Nurser 3 grow lights, but you can really use any sort of LED lights. My lights that you see behind me are a little bit too far from the plants. When I move laboratories in June, I'm moving into a larger space. Spoiler alert. I'm gonna
She has a
rearrange
laboratory. That's cool. Jules, yes. You study infectious disease, full-time microbiologist. Okay, yeah, you gotta stay safe away from those beasts in your biosafety cabinet.
shelves so that the plants sit about like 4 to 6 inches higher than they're currently sitting here. The goal
That's all you
after
need, 4 to 6
weeks
inches.
of the explant being on the establishment media is to see some shoot formation. Here are some examples of what shoots will look like on various types of tropical plants. Each shoot is going to form into a new plant, so ideally you want multiple shoots to form so you end up with a lot of plants. If you only get one shoot, that's okay, don't worry, more will form later. So yeah, the protocol says after 6 weeks, subculture the shoots onto multiplication media. Subculturing, which I was just talking about, is the process of transferring the plants or the explant from one container of media and putting it into a fresh container of media. You would keep subculturing the shoots and eventually the baby plants every 6 weeks or so.
Various tropical pothos. Plants. Pothos. Yeah. The aeroids. Yeah. Variegated and unique cultivated varieties of aeroids is what she showed. Why doesn't she move the racks? It's annoying to move them. I don't blame her. But yeah, you could move the racks.
For about 3 to 5 or 6 months until you had the desired quantity of plants. Once you have the desired amount of plants from the multiplication stage, then you would prepare the rooting media and transfer your baby plants onto the rooting media so that they can grow larger before they're ready to come out of tissue culture. I want to show you guys how to set up for a subculture inside of the still air box. I'm using a different type of plant for the subculturing demonstration. A better YouTuber would have had the monsteras ready to go. I'm trying my best. Essentially, to do a subculture inside of a still air box, you need your plants, fresh tissue culture media that you have pre-prepared, a glass bead sterilizer, and a pair of sterile forceps. I start each sterile transfer by cracking open the lid of the fresh container of media. I leave the lid resting on top so that no airborne contamination can get inside the container. I open the other container that holds my baby begonia plants. Then, using the sterile forceps, I remove the plants from the old media and place them into the fresh tissue culture media. I never place the lid of a container of fresh media down. I keep it in my left hand as I handle the plants with my forceps. I try to be very efficient inside the still air box to try to prevent contamination as much as possible. No matter what I do, I will get people in my comments saying that you can't use a still air box for tissue culture, that it doesn't work, that you need a laminar flow hood, and none of that is true. If you're curious,
I mean, in her laboratory, she probably does have a laminar flow hood. I mean, looks pretty legit.
laminar flow hood, like the one you can see
she
behind
has
me,
one behind her.
blows air through a HEPA filter to create a really clean work surface. And yes, it is more effective than a still air box at preventing contamination for that reason, but the still air box still works. The begonias that I just showed were subcultured on January.
Spray the air and hold your breath. Stream of May. That's your technique. That's your tried and true technique. Duskborn, you said this makes me think of those pet memes. You bought a purebred and I found mine in the garbage. Yeah. The thing is that the output will be the same thing. Well, yeah. You might have minor morphological differences at like the base of the plant between if it was grown from seed or cutting. But yeah. Yeah, string bean. Green bean stream. You think of the person with the kind of money, probably. Yeah, $2,000 for a flow hood. Might as well if you've already invested so much, right?
2nd. Today is March 5th. And how many of those begonias got contaminated? None of them. They're right here. And actually, they are desperately due for another subculture. It has been way too long. I actually paid $350 for the begonia that I originally used as the tissue sample to produce all of those clones. So not only is their existence a testament to the success of the Still Air box, but also they're a great example of how you can actually make money doing tissue culture. The biggest pitfall that you will encounter during the TC process is contamination. Here are some different examples of what contamination can look like. My personal favorite contaminant is slime mold. Leave yours in the comments below. Contamination is caused either by
Slime mold. Slime mold. You know that Monsanto hates her? I don't think Monsanto hates her. I think it's Palm Street. I think it's the people who have those like rare plants they're trying to sell for $1,000. Those are the people who hate her. I don't think Monsanto cares. They're not in the same industry. Raise your hand if you're a slime mold. What is the Twitter meme with the dog that's like peeing with the music? That's a slime mold music. I want to watch this dog urinating while oscillating. Give it to me. Where is that dog? I can't find it. Anyway, you know what I'm talking about. Add antibiotics. Well, that won't stop slime molds, right? Prima Bay. It'll stop bacteria. Yeah, it doesn't work on fungus. It'll help though. The cultivar populations of plants is realistically affecting cultivars of aroids and begonias. Yeah. Those are the ones that people are, selling for so much. One person can only do so much. I think she's doing great education of some field, that she's democratizing a scientific field that is sometimes inapproachable, creating accessible protocol. And she's specifically targeting houseplants, which I think is great. The awareness is really good, if anything else. Yeah. I think the good thing is that she is helping people learn and grow and teaching people that, destroying that strange houseplant industry is quite good, And then people can take this technique and apply it to other things. So if someone is obsessed with native plants of Georgia, then they can learn this. Start it with your pothos, practice on your pothos, and then do Georgian native plants. Do you think she has plant mules that get her rare plants because they won't sell it to her? Oh, Duskborn, surely. If she needs someone, let me know. I can do it. I should tell her I can do it. They don't need to be poached from the wild. True, great point. Yeah, true. Plants grown by tissue culture don't need to be poached from the wild. Yeah. So the ones that she's doing are like rare cultivated varieties and hybrids. And by rare, I mean there's not many of them and people want a bunch of them. But yeah, cultivate critically endangered plants. And the thing too is that they might have different requirements. They might have different requirements for like the ratios. So she gave the ratios for Monstera, and a lot of these things are well researched. Carnivorous plants have been poached. Oh my gosh, yeah. That makes sense. At UC Berkeley, they sell a lot of carnivorous plants. And I remember 10 years ago, I went into a lab and I was like, what's going on here? They said, we're doing tissue culture of Venus flytrap. I was like, what? They said, yeah, we take the leaf and it works that way. And so they did. I mean, I never seen people tissue culturing Venus flytrap, but there are a bunch of old ladies doing it. The docents, The trouble with rare Australian plants, they're rare because they grow slowly. So not sure if it'll help. That makes sense. Green Bean Stream, you're a carnivorous plant specialist. Really? Okay. Green Bean Stream and True, you're both carnivorous plant people. We have a friend in YouTube from Europe. Good morning. Breakfast video. Welcome in. We're talking about tissue culture, which is where you take a little piece of a plant and you grow it in a small container with like a gelatin. It's not gelatin, it's called agar, but it's like jello. And then you grow it up and you have a small plant. They're clones. So with this particular practice, you know, you can do a lot. And it's quite challenging with contamination. So she's talking about contaminations.
Not cleaning the explant thoroughly enough before it goes into the tissue culture media, or just having poor sterile technique practices. Unfortunately, contamination will cause your plants to die. Contamination is pretty much inevitably going to happen when you are new to doing tissue culture and using sterile technique. That's why I always tell people to start by experimenting on normie ass monsteras like the one we use today, instead of using your single node of your Kunzo lineage.
So these types of things I really don't care for, because sometimes, you know, people mean very well and they say, "Oh, Matthew, do you have any favorite rare plant?" And they're thinking this type of thing. Kunzo is like I guess he's a Japanese aroid grower, but this particular species with this particular name that's variegated and all this, you know, I'm not, I really don't care about them. They're not that exciting. I don't even like variegated plants. But I like cool plants that have interesting features. Often, sometimes those are rare. Rarity is, there's different types of rarity. And I think this lady has a video on that too. Oh, Relo, have a good night. Take care. Go to your bed and kitty cat. All right.
Philodendron ilsmanii. If your culture does become contaminated, you have two options. Option 1. Start over. Repeat the experiment, but this time either increase the bleach concentration or the amount of time that you leave the explant in the sterilization solution. Option 2 is to re-sterilize the explants, take them out of the contaminated media, re-sterilize them, and try to put them back into tissue culture, basically repeating the entire establishment stage of tissue culture. If you are struggling with contamination and feeling frustrated, please post about it on the Plants in Jars Discord server. If you can describe the
I think this is probably the clearest I've ever seen someone describe tissue culture. So, Jules, I agree. All those rare plants look the same to you? There's little tiny changes, little nuance, you know? Yeah. If you're struggling with contamination, wash your hands, blow your nose. Have you considered adding in a mutagen and trying to make it grow weird features? Interesting.
That you followed and include photos. We can usually figure out what went wrong and help you prevent contamination in the future and improve your sterile technique. The link to join the Discord server will be in the description below. One alternative to going through the entire establishment process is to just start with a plant that's already in tissue culture. For example, in this video from last year, I started with a Philodendron Red Anderson that I got in tissue culture from a plant laboratory in Thailand. Using the same exact still air box I just showed you for the demonstration, I transferred that plant into multiplication media. Over time, that plant grew and multiplied, and now I have hundreds, if not thousands, of clones of that plant that was started in the still air box. So it is a great way to start doing tissue culture, and another W for the still air box. I personally think that method that I just described is the easiest way to get started doing tissue culture. Welcome to the Q&A portion of my video.
Discord needs an introduce a crossover episode. I want to meet their Discord users with delegation of our own. Yeah. Our Discord will meet their Discord. What did that method eliminate? Cutting the plant? Which method? The one she described?
Q&A, I will be posing questions to myself and also inserting them. Question 1. Why not just grow plants from seed?
Okay.
At the very beginning of the video, I mentioned that plants can reproduce both sexually and asexually. Sexual reproduction in plants involves flowers and pollen. Pollinators like birds and bees transfer pollen between flowers, resulting in seeds that eventually grow into new plants. These plants that are grown from seed are going to have traits from both parent plants. For example, my friend Justin from Created in a Garden on Instagram crossed 2 different anthuriums, and each of the seedlings displayed unique leaf shapes and sizes due to this genetic mixing, despite having the same exact parents. Genetic diversity is very cool. Crossing different plants together can
So that point was, I think, the key to it all, is that here there's 2 distinct parents that gave a bunch of seeds, and you grow all the seeds and every kid looks different. You don't look like your brother and you don't look like your sister. What if you could clone your brother? Then you'd have a bunch of yourself, or your brother, not yourself. Such a pain. Funny how she used birds and bees as pollinators examples for sexual reproduction. That's right, little birds and bees.
sizes due to this genetic mixing, despite having the same exact parents. Genetic diversity is very cool. Crossing different plants together can result in really cool new hybrids, like this begonia hybrid from China that I'm currently obsessed with. But for commercial plant sellers, there is usually value in uniformity. Asexual reproduction methods produce genetically identical plants. Plants can be propagated asexually very simply without tissue culture by taking a cutting or cutting a part off a plant and allowing it to grow into a new plant. That new plant will still be a clone of the original plant that it was taken as a cutting from. Doing tissue culture is also an asexual method of reproduction. It's actually very similar to taking a cutting, but in TC, we're placing our cutting onto a nutrient-rich culture medium, so it has the optimal conditions to grow and to multiply. Question 2.
We taught this one class. It was like a college class on propagation, and it was basically like, hey, we're going to teach you everything you would need to know about plant propagation so that when you go into horticultural industry, you will know what you need to know. And basically, the professor concluded that you never need to learn tissue culture until it is like the last resort because of its amount of steps, effort, and sterile technique and contamination. And you have to harden the plants off too. I mean, there's lots of little things to consider. So, yeah. If you can do stem cutting and leaf cutting and root cutting or suckers or anything else, do that before you do tissue culture.
What is the difference between shoot culture and callus culture? There are multiple types of tissue culture you can do. Shoot culture is by far the easiest. It's also by far the method that I do the most in my own laboratory. In shoot culture, which is also called direct organogenesis, if you want to sound super smart, we're getting the plants to grow directly from the explant. One other form of tissue culture that I want to talk about, because I get a lot of questions about, is called callus culture or indirect organogenesis. In callus culture, we're trying to grow callus first.
So this is what I always considered the callus version, because this is what I had seen in laboratories. Because in my experience, typically if you can grow it from a node, you might as well do a stem cutting or like you might as well not do this type of propagation if you can grow it from a node.
Instead of shoots. A callus is just a mass of undifferentiated cells. It just looks sort of like a clump. It can be green, it can be brown, it can be yellowish white, but it's always a clump. There are a few ways to get callus to form from an explant. One way is to use a very strong synthetic plant growth regulator in the media, like 2,4-D. Another way is to use a cytokinin, like BAP, and an auxin, like NAA, in an equal ratio. That can sometimes result in callus as well. Here is a cheat sheet for cytokinin.
Yes, okay. This is like a foundational plant physiology for plant growth regulators. Mad Maxine It looks like plastic. It does. Yeah. If there's so cytokinins and auxins are both plant growth regulators, like chemicals that alter growth and development, and they often interact in a way that is antagonistic. So if there's more cytokinins than auxin in a certain tissue, then it'll start making a bunch of leaves and stems. If they're about the same, it'll become a blob of undifferentiated things. And so when you're growing these, you want to make sure that hormones are powerful magic, yeah. You want to make sure you understand the ratios. If you have more auxin cytokinin, it's roots. Dalalok, I'm not sure what you said.
To auxin ratios and their outcomes. After the callus forms, when you're doing callus culture, you would transfer the callus to a shooting medium and try to get baby plants to form directly out of the callus. Callus culture sucks, in my opinion, sorry, because you need to be subculturing it like every 2-3-ish weeks instead of like 6 to 8 for shoot culture. So I just personally find it a huge inconvenience. I also really don't like using very strong synthetic plant growth regulators. Callus culture is a common way to grow anthuriums in tissue culture. Like I said, it's a lot of work.
Yeah, that's my understanding. The people I knew who did tissue culture in the lab were all anthurium people. And they would take little pieces of leaves, and you get a whole plant from a piece of a leaf. Isn't that crazy? It's not even meristematic, but they create meristems, basically.
Shoot culture is much, much easier, and that is why you don't see me working with a lot of anthuriums. My favorite question, can you make money doing tissue culture? So yes, you can make money doing tissue culture as a service, or by selling TC plants as products. Actually, if you want to have a business selling plants that is scalable, then I will take it a step further and say you need to be doing tissue culture in some capacity. Otherwise, you're going to be very at the whim of supplier stock. Please keep in mind, these are just my opinions. I have sold plants in the past, but never at like a commercial level, okay? My advice is also somewhat skewed toward tropical ornamentals, since those are the plants that I enjoy and have worked with a lot in the past. My first tip is to focus on one specific genus and get really good at it, really specialize.
Very good tip.
That way you'll be able to use the same protocols over and over and really perfect them, and even eventually develop your own proprietary protocols for tissue culturing that specific type of plant. My second tip.
Why does she do this? Why does anyone do anything? I think she likes it.
This one might seem obvious, but stay up to date on plant trends. Yes, plants are not as trendy as they were during COVID, but there still are a lot of people collecting plants as a hobby. I would say that 2024 was like the year of foliagey anthuriums. Personally, I really want gymnos to be a thing, particularly these really cool ones being produced in Thailand right now. I think they would also do really well in tissue culture. I've tried to buy them, but the guy will only ship them to Thailand, unfortunately. My last tip for selling plants is to either exclusively work with non-variegated plants or only plants that have very stable variegation. A lot of plants with unstable variegation, like the variegated Boletiae, will almost always revert to the non-variegated version in tissue culture or once you take them out of TC. And most of the time, the non-variegated versions of these plants are much less valuable than the variegated versions. I mentioned also that tissue culture can be a service. Orchid flasking is another way to make money by doing TC. Orchid flasking is a service where a customer will send you a green seed pod or dry seeds and ask you to germinate them and grow them and then return the seedlings after 6 months to a year. Orchid seeds are really, really small, basically like dust, so the only way to germinate them is through tissue culture because these seeds have no energy reserves of their own. Nick Rust from Rusty Exotics recently sent me
that's the guy.
some orchid.
Yeah, Rusty Exotics. If you want to see someone who does this quite expertly on the internet, go find him, Rusty Exotics. He had a video recently where he like deflasked a bunch of orchids, and it's like crazy. They can't be true though? Wait, what? Green bean stream, what can't be true? Orchid flasking sounds like a euphemism. He's a bit of an orchid flasker. He's a bit of a nighttime botanist. He's a bit of a homosexual. He's a bit of a fairy boy.
of a catasetum and also a habenaria. So we put those into tissue culture. So I should have some orchid content coming soon for the orchid enthusiasts in the house. How do you store tissue culture chemicals? So the storage instructions are always going to be printed on the bottle. Agar and other gelling agents can just be stored at room temperature. MS needs to be refrigerated.
Justin, the horse-like ladies, that's right. How are you doing?
And plant growth regulators, it depends. The BAP and NAA get stored in the refrigerator, and then the IBA should actually be stored in the freezer. IBA will freeze solid in the freezer as a solution. So if you're going to take a long time to use all 100 milliliters of the IBA solution, it makes sense to separate it into 25 milliliter aliquots. One aliquot can get stored in the fridge for usage, and then the other ones can get stored in the freezer so that they'll last longer. This is true for all IBA solutions. I have never heard another chemical seller tell people that. I think because it seems inconvenient, but I want to set you guys up for the most success possible. What is the easiest plant to try to tissue?
Okay, who at this point is interested in doing tissue culture? We've seen a lot. We've seen a lot from her, everyone. Forte, hello, good to see you. We're streaming both on YouTube and on Twitch. And so we have a lot of folks in Twitch that we've seen for a while. Forte, hello. Green Bean Stream. You can understand wanting to higher yield percent from your orchid seeds, but if tissue culture was the only way to get a good yield, but saying they don't yield without TC. You don't want to tissue culture plants like this. Yeah, I would not do this unless it was a last resort. Justin, I'm doing quite well. Having a fun time. Mad Maxine, you might try it? Okay. Sable, you're... Yeah, do it on your university's dime. You have your own thoughts about it. How you would prep some sterilization. Oh yeah, okay, Ananoa, you're a professional. You're do you want to share some of the things you would do that would be different than what she described? Forte, it's never too late. If a company wanted to hire you to do tissue culture or something, you would do it. Yeah, I mean, I would be the same way. If someone said, hey, we'll give you a livable wage to do this for 20 hours a week or 15 hours a week, I would, I guess that wouldn't make sense. Livable wage for 20 hours a week, but I would do it. But only for 15, 20 hours a week. Wouldn't do it any more than that. I wouldn't be outside with the dirt. Livable wage 20 hours a week does make sense. Really? Okay. If someone gave me 3 wishes, my first wish, have a livable wage working 20 hours a week, doing tissue culture. Okay, Forte, we'll open up the server. Better position than pulling weeds in a hothouse all day? Wish number 2, peace and love on planet Earth. And wish number 3, complete and total annihilation of the Apiaceae. Those are my 3 wishes. You love pouring agar plates for bacterial growth, fungal growth. So you would prep the agar, autoclave that, and pour out the media into containers in laminar flow hood cabinet. Okay. Maxine, your entomology background turned you into being outdoors in the woods with ticks. Oh, arthropods? That's okay. We need arthropods. We don't need Apiaceae. I didn't call for a war. Hold on. I said peace on earth for all people. I said peace and love. That wish number 2 would absolve me of the issues of wish number 3.
first. I think the easiest plants to experiment on are African violets. You see results really fast, so I think they're great for beginners, and they're also really cheap plants to buy, so you're not out a bunch of money if it gets contaminated. Philodendrons are another.
African violets. Forte. Number 1, Petco where the pets go. Number 2, African violets. The old lady plant. I was the only guy, well, I was one of the few male presenting individuals in the African Violet Club. I need to get some. It would be good. African violets. It's the right thing to do. Okay, let's see. She said philodendron for number 2. Let's finish this. We're almost done.
One that is great to experiment with when you're first getting started. I think they're really fun to grow in tissue culture as well and not that difficult. I have protocols for both of those types of plants in the Plants in Jars protocol library to help you get started. Where can I learn more about tissue culture? Great question. The biggest problem that I had when I was first getting started was that there was just no information available, like anywhere. At least not a lot of accessible information that I could easily understand as someone without a science or laboratory background. I don't think I mentioned this earlier in the video, but I majored in finance in college, and then after that, I worked for a real estate developer, kind of doing project management type stuff. One book that is an amazing resource, which is also doubling as my microphone stand currently, Plants from Test Tubes. The way that this book is written is very accessible to a layperson without a lot of background knowledge, so I think it is such a fantastic resource. Read it front to back. For any specific questions, I would say that my Discord channel is also a fantastic resource. We are the largest tissue.
Okay, if you want to join, there's the link for her Discord. Your mom, Sable Miner, used to grow a ton of African violets back in the 70s. Cute! For folks who don't know, we'll look at the African violets are one of my favorite groups of plants. And I had a bunch when I was in Austin. But they're charming. And you can put them in self-watering pots. They're quite nice. They look like this. Lots of different flower colors, shapes. Anyway. Well. Yeah. Changing Wish 3 to reviving the rare plant you mistook for a snake plant. Oh, Justin. Okay, so Justin, he was in the propagation class that I helped TA. And I forgot about that. Yeah. We told you to go and cut a plant. And you went and cut a plant, but it wasn't clear which one to cut. And you cut a very rare, slow-growing plant. That was good. I think it was the red sealing wax palm. I think this is the one you cut. I'm going to pull it up. I think we were trying to tell you to cut the snake plant. And our friend Justin cut this one instead, which is extremely slow growing. It's not rare, it just grows slowly, and you can't propagate it from, like, pieces of the stem. But it's beautiful palm. Anyway. Well. It was the most expensive plant that we had in the greenhouse. It was funny. Well, seal of approval is here. And the seal of approval is curious to answer Duskborn's question. Duskborn had a question about Terra. No, the Murashigi and Sku mix. Seal of approval. Do you approve the MS Media? Says yes. Heavily approves of the MS Media. Great. It's filled with micronutrients. It has micro and macronutrients and a bunch of other things that I need to figure out what they do. There's vitamins, additional compounds. Well, folks, I think that is where we will end it for today. Yeah, the seal of approval. Approved of the MS Media. After all, a round of applause. We need to learn more things about the Earth. True. That is a fact. We need to learn everything. All right, well, we will see you tomorrow. Have a good night. Jules, Ananoag, True, Duskborn, Justin, Elfingor, Salty Boy. Have a good night. Ungoy. Of course, you miss everything. We'll be here tomorrow. I don't know what we're going to talk about, but we'll be here tomorrow. Sable Miner, take care. We have a lot of friends. Well, we have a growing group of friends on YouTube, too. And so this stream, if you want to watch it, it'll be on YouTube. It'll also be on Twitch. All right. The Grove grows. Yeah. Bye.